So today our topic is hemoglobin and iron metabolism. First introduction. Hemoglobin is iron containing coloring matter of red blood cells. Red color of red blood cells is due to hemoglobin. So it is a chromoprotein forming 95% of dry weight of red blood cells and 30 to 34% of wet weight. Now, molecular weight of hemoglobin is 68,000. Now we will see normal hemoglobin content. Average hemoglobin content is 14 to 16 gram per deciliter. Varies depending upon age and sex. So first, age. At birth, 25 gram per deciliter, highest. After third month, 20 gram per deciliter. So hemoglobin count decrease. Why? Because of decrease in RBC count. After 1 year, 17 gram per deciliter. From puberty onwards, 14 to 16 gram per deciliter. At birth, hemoglobin content very high because of increased number of red blood cells. Then due to physiological jaundice, red blood cells count decrease. Sex. In adults male, 15 gram per deciliter and in adult female, 14.5 gram per deciliter. Why low in females? Because of menstruation. There is blood loss in menstruation. Now we will see functions of hemoglobin. First, transport of respiratory gases. So, oxygen from lungs to all tissues. Then, second is carbon dioxide from tissues to lungs. First is transport of oxygen. When oxygen binds with hemoglobin, a physical process called oxygenation occurs, resulting in formation of oxyhemoglobin. So first oxygen bind with hemoglobin, resulting in formation of oxyhemoglobin. Okay. Iron remains in ferrous state in this compound. So oxyhemoglobin में iron कैसा होता है? So in ferrous state. Oxyhemoglobin is an unstable compound and combination is reversible. That is, when more oxygen is available, it combines with hemoglobin, and whenever oxygen is required, hemoglobin can release oxygen radially. Okay. When oxygen is released from oxyhemoglobin, it is called as reduced hemoglobin or ferrohemoglobin, because in this compound iron is in ferrous state. Transport of carbon dioxide. Carbon dioxide binds with hemoglobin, then this compound is called as carb hemoglobin. Also unstable and reversible. Affinity of hemoglobin for CO2 is 20 times more than oxygen. Now, second function is buffer action. Hemoglobin acts as a buffer and play an important role in acid-base balance. Okay. So, second function is buffer action. Now we will see structure of hemoglobin. So first, what is hemoglobin? Hemoglobin is conjugated protein. Consists of protein combined with an iron containing pigment. So what is hemoglobin? Hemoglobin is a conjugated protein, consists of protein combined with an iron containing pigment. Protein part is called as globin and pigment is heme. In heme there is iron. Okay. Heme also forms a part of structure of myoglobin. So what is myo? Myo means muscle. Okay. Oxygen binding pigment in muscle. And neuroglobin. Neuro means related to brain. Oxygen binding pigment in brain. Now we will see first iron, then porphyrin, then globin. First iron, present in ferrous form. It is unstable or loose form. In some abnormal conditions, iron is converted into ferric state. So stable form. So usually in hemoglobin, iron present in ferrous form. it is unstable or loose. But in subabnormal condition, iron will become stable. That means ferrous is converted into ferric state. Then porphyrin. We already seen what is porphyrin in destruction of RBC. So what is porphyrin? Pigment part of hemoglobin formed by 4 pylor rings called as tetrapylor, called 1, 2, 3, 4. Okay. Pylor rings are attached to one another by methane bridges. Okay. So one pylor ring attached to another pylor by methane. Iron is attached to end of each pylor ring. So N of globin molecule. So iron is attached to N of each pylor ring and N of globin molecule. So what is globin? So globin contains 4 polypeptide chain. Among 4, 2 alpha chain and 2 beta chain. Now we will see types of normal hemoglobin. So 2 types. First, adult hemoglobin. Second, fetal hemoglobin. Replacement of fetal hemoglobin by adult hemoglobin start immediately after birth, completed about 10 to 12th week after birth. First we will see what is structural difference between adult and fetal hemoglobin. In adult hemoglobin, globin contains 2 alpha chains and 2 beta chains. Clear? In adult hemoglobin, globin contains 2 alpha chains and 2 beta chains. In fetal hemoglobins, there are 2 alpha chains and 2 gamma chains. Okay. And now, what is functional difference? So, fetal hemoglobin has more affinity for oxygen than that of adult. Okay. So, fetal hemoglobin have more affinity for oxygen than that of adult. So, now we will see abnormal hemoglobin. Pathological mutant forms of hemoglobin. This variant are produced because of structural changes in polypeptide chains. These variants are produced due to structural changes in polypeptide chains caused by mutation in genes of globin chains. So, in this usually there is mutation in globin chains. There is 2 categories of abnormal hemoglobin. First is hemoglobinopathies. Second, hemoglobin in thalassemia and related disorders. So, first we will see hemoglobinopathies. What is hemoglobinopathies? So, it is genetic disorder caused by abnormal polypeptide chains of hemoglobin. So, first is hemoglobin S, found in sickle cell anemia. Hemoglobin S found in sickle cell anemia. Alpha chains are normal and beta chains are abnormal. Okay, clear? So, second is hemoglobin C. Beta chains are abnormal and alpha chains are normal. Found in people with hemoglobin C disease, which is characterized by mild hemolytic anemia and splenomegaly. Then third is hemoglobin E. Beta chains are abnormal, present in people with hemoglobin E disease, usually characterized by mild hemolytic anemia and splenomegaly. Then fourth is hemoglobin M. In this, there is abnormalities in both chains, alpha and beta. Okay. Abnormal hemoglobin present in form of methemoglobin occurs due to mutation of genes of both in alpha and beta chains, resulting in abnormal replacement of amino acid. Present in babies affected by hemoglobin M disease or blue baby syndrome. Then second is hemoglobin in thalassemia and related disorder. In thalassemia, different types of abnormal hemoglobin present. In alpha thalassemia, there are alpha chains are reduced, absent or abnormal. And in beta thalassemia, beta chains are reduced, abnormal or absent. Now we will see abnormal hemoglobin derivatives. Hemoglobin derivatives refer to blood tests to detect and measure percentage of abnormal hemoglobin derivative. Okay, so abnormal hemoglobin derivative are formed by carbon monoxide poisoning or due to some drugs like nitrates and sulfonamide. Okay, so first abnormal hemoglobin derivatives are first carboxyhemoglobin, second methemoglobin, third sulfhemoglobin. We will see detail one by one. Normal percentage of hemoglobin derivatives in total blood. So this percentage are normal in normal people. So carboxyhemoglobin 3 to 5%, methemoglobin less than 3%, sulfhemoglobin is negligible. Okay, so first we will see the detail of carboxyhemoglobin. First, combination of carbon monoxide with hemoglobin called as carboxyhemoglobin. Carbon monoxide is colorless and odorless gas. Hemoglobin has 200 times more affinity for carbon monoxide than oxygen. So, normal hemoglobin has 200 times more affinity for carbon monoxide than oxygen. It hinders transport of oxygen, so resulting in hypoxia. Okay. If hemoglobin is not combined with oxygen, then in all our body there will be less O2 supply. So it will result into hypoxia. Now, sources of carbon monoxide: charcoal burning, coal mines, deep wells, drainage system, exhaust of gasoline engines, gases from guns and other weapons, heating system with poor or improper ventilation, smoke from fire, and ninth, tobacco smoking. Clear? So this is source of carbon monoxide. People working in this industry usually having carboxyhemoglobin more percentage than a normal person. Sign and symptoms of carbon monoxide poisoning. So, while breathing air with less than 1% of carbon monoxide, Hb saturation is 15 to 20%. Less than 1% of carbon monoxide, so hemoglobin saturation is 15 to 20%, and mild symptoms like headache and nausea while breathing air with only less than 1%. While breathing air more than 1% of carbon monoxide, Hb saturation is 30 to 40%, causes severe symptoms like convulsion, cardiorespiratory arrest, unconsciousness and coma. When hemoglobin saturation increase above 50, so usually death will occur. So clear about carboxyhemoglobin. Now second is methemoglobin. Abnormal hemoglobin derivative formed when iron molecule of hemoglobin is oxidized from ferrous state to ferric state, called as methemoglobin. Okay. Methemoglobin is also called as ferrihemoglobin. Now we will see, under normal circumstances also body faces threat of continuous production of methemoglobin, but counteract by erythrocyte protective system called as nicotinamide adenine dinucleotide system, which operates through two enzymes. Clear? Under normal circumstances also body face continuous threat of production of methemoglobin, but counteract by erythrocyte protective system called as nicotinamide adenine dinucleotide system, which operates through times. First is diaphorase 1, nicotinamide adenine dinucleotide phosphate dependent reductase, okay, responsible for 90% of our action. Then diaphorase 2, NADHP dependent methioglobin reductase responsible for only 5%. Now we will see self-hemoglobin. First is abnormal hemoglobin derivative formed by combination of hemoglobin with hydrogen sulfide, usually caused by drugs such as phenacetin and or sulfonamide. Self-hemoglobin cannot be converted back into normal hemoglobin. Only way to get rid, then RBC will complete their lifespan and destroyed. Okay, RBCs with self-hemoglobin destroyed after their lifespan. In blood level, very negligible. So in normal person, there is negligible amount of self-hemoglobin. But when its level rise above 10 gram per deciliter, cyanosis will occur. Usually serious toxic effect. Okay. So now we'll see synthesis of hemoglobin. Actually start in pro-erythroblastic stage. अगर आपको नहीं पता pro-erythroblastic stage क्या होता है, so पहले आप erythropoiesis and stages of erythropoiesis पढ़ोगे तो आपको पता चलेगा कि pro-erythroblastic stage क्या था. So हमने देखा था pro-erythroblastic क्या फर्स्ट स्टेज था. So actually start in synthesis of hemoglobin actually start in pro-erythroblastic stage but appears in intermediate normoblastic stage. Heme portion of hemoglobin synthesized in mitochondria and globin in ribosome, and heme and globin together will form hemoglobin. Okay, so first we will see mitochondria. So this is mitochondria. In mitochondria, succinyl CoA plus glycine together form amino-levulinic acid with the help of ALA synthase. What is ALA? So amino-levulinic acid. Then two molecules of amino-levulinic acid goes in cytoplasm. Two molecules convert into porphobilinogen with the help of ALA dehydrase. Clear? Then porphobilinogen convert into uroporphobilinogen 1 with the help of synthase. Then uroporphobilinogen 1 convert into uroporphobilinogen 3 with the help of co-synthase. Then in last, uroporphobilinogen 3 convert into porphyrinogen 3. Then porphyrinogen 3 goes again into mitochondria. Then it oxidizes to form protoporphyrinogen. Then it oxidizes to form protoporphyrin. Then with the help of iron in ferrous state, it convert into heme with the help of ferrochelatase. Clear? Then heme plus globin together will form hemoglobin. So this was synthesis of heme. Now we will see synthesis of globin. Okay. So formation of globin. Polypeptide chains of globin are produced in ribosome. Already we seen. 4 types of polypeptide chains, namely alpha, beta, gamma and delta chains. Each globin for a molecule is formed by a combination of 2 pairs of chains. So each globin is formed by a combination of 2 pairs of chains, and each chain is made up of 141 to 146 amino acid. So each globin is formed by a combination of 2 pairs of chains. Then each chain is made up of 141 to 146 amino acid. Then we already seen that adult hemoglobin contains 2 alpha chains and 2 beta chains, and fetal hemoglobin contains 2 alpha chains and 2 gamma chains. So this was formation of globin. Then globin from ribosomes and heme plus heme from mitochondria together will form hemoglobin. Heme plus globin together will form hemoglobin. So clear? So now we'll see iron metabolism. First we will see importance of iron. Iron is an essential mineral and an important component of proteins involved in oxygen transport. So, it is mineral and an important component of proteins involved in oxygen transport. Important for formation of hemoglobin and myoglobin. So, important in formation of hemoglobin and myoglobin. Iron also necessary for formation of other substance like cytochrome, cytochrome oxidase, peroxidase and catalase. Okay. Now, what is normal value and distribution of iron in body? So, total quantity of iron in body is 4 gram. In hemoglobin, it is 65 to 68 percentage, highest percentage. Okay. In muscle as myoglobin 4 percentage. As intracellular oxidative heme compound 1%. In plasma as transferrin 0.1%. Stored in RE system 25 to 30 percentage. Clear? Then dietary iron. So available in two forms, heme and non-heme. First we will see heme iron. Present in fish, meat, chicken. Heme iron is absorbed easily from intestine. So first heme iron, then non-heme iron. Available in veggies, greens and cereals. Not absorbed easily from intestine. So now we will see absorption of iron. First, absorbed mainly from small intestine. Absorbed through intestinal cells by pinocytosis and transported into blood. First, we'll take iron from diet, then it is absorbed from small intestine. After absorption, it is transported into blood. Bile is essential for absorption of iron. Okay, bile is essential for absorption of iron. Iron is present mostly in ferric form. Okay. It is converted into ferrous form which is absorbed into blood. HCl from gastric juice makes ferrous iron soluble. So it could convert it into ferric iron by enzyme ferric reductase from enterocytes. From enterocytes, ferric iron is transported into blood by protein called ferritin. Okay. In blood, ferric iron is converted into ferrous iron and transported. So now we'll see transportation of iron. Immediately after absorption into blood, iron combines with beta globulin and called as apotransferrin, resulting in formation of transferrin. Okay. Iron is transported in blood in form of transferrin. So iron is transported in blood in form of transferrin. Iron combines loosely with globin and can be released at any region of body where iron is needed. Okay. Storage of iron. Stored in large quantities in RE cells and hepatocyte. In cytoplasm of cell, iron is stored as ferritin in large amount and in small quantity as hemosiderin. Okay. Usually excreted. Now we will see daily loss of iron. In males, about 1 mg of iron is excreted every day through feces. In females, the amount of iron loss is very much high. Why? Because of menstruation. Clear? Then 1 gram of hemoglobin contains 3.34 mg of iron. Clear? 1 gram of hemoglobin contains 3.34 mg of iron. 100 ml of blood contain 15 gram of hemoglobin and about 50 mg of iron. How? So 1 gram of hemoglobin contain 3.34 mg. So 3.34 mg into 15 gram. So 50 gram of iron. So if 100 ml of blood is lost from body, there is loss of about 50 mg of iron. Clear? In females, during every menstruation cycle, about 50 ml of blood is lost, by which 25 mg of iron is lost. Clear? This is why iron content is always less in females than males. Iron is lost during hemorrhage and blood donation also. Okay, if 450 ml of blood is donated, about 225 mg of iron is lost. So here we completed synthesis of hemoglobin and iron metabolism. So thank you for watching.